Center for Clinical Trial. Okay. To the first clinical trials and evidence synthesis seminar of the year. I'm delighted to have Richard Chaisson speaking today. I've known Richard since he arrived at Hopkins a few years ago as then a hotshot AIDS researcher. Over the years, he's continued to be a highly productive AIDS researcher, but he's also one of, or if not the world's experts in tuberculosis. He's a product originally of the University of Massachusetts School of Medicine. He did most of his training, or all of his training pretty much at the University of California, San Francisco, where he started out on the faculty. He came to Hopkins in 1988 and rose rapidly to the rank of professor. He is a professor of medicine, epidemiology, and international health. He is an incredibly prolific author with 360 original publications, 113 reviews, and 65 book chapters. He advises both the U.S. Public Health Service and the WHO on AIDS and tuberculosis. And he has been involved in several critical trials on the treatment of tuberculosis. And he's going to talk to us today about progress and challenges in clinical trials for tuberculosis. Dick, thanks for coming.
Thank you, Doug. Delighted to be here. I'm going to talk about TB clinical trials. I'm going to try to cover a lot of territory, and I'm giving the perspective of someone who does clinical trials, but from the clinic perspective. There's a lot of methodologic issues that I'll touch on, but I want to make it clear I'm not a methodologist, so I'll touch on them because I think they're important, and maybe we can discuss them later. Oops, I've got to find which button to work. Okay, I need to make these disclosures about funding and relationships. And I want, particularly for this audience, just to give a little important background on tuberculosis. So TB is an airborne infectious respiratory pathogen, and it causes about 10 million cases per year and 1.5 million deaths. It's the leading infectious cause of death in the world, currently still ahead of COVID. HIV-related TB accounts for about 10% of cases and closer to 20% of deaths. And multi-drug resistant or rifampin-resistant tuberculosis is an important global problem, about 5% of the cases and a disproportionate fatality rate. So this is the backdrop, and just to show you that TB has eclipsed HIV as the leading infectious cause of death, and it's the most important cause of death in people with HIV. So TB is a disease that has an unusual life cycle in humans. It's spread human to human. There's really no environmental reservoir. After infection is acquired, people can rapidly progress to tuberculosis disease, and that's primary TB, or they can have latent tuberculosis that varies in the degree of pathology that it causes, but can later come out to cause clinical tuberculosis. But from a clinical perspective and a clinical trials perspective, we really have two things. We have active tuberculosis disease and we have latent TB infection, and we can treat both the active TB to prevent death and reduce transmission and make people better, and latent TB so they never become cases. And so that's what we'll focus on. Now, the reason that I'm talking about both is that all of the mathematical modeling, this is probably the simplest and best mathematical model that's out there for controlling TB globally, but they all show the same thing, that better treatment of active TB and massive expansion of the treatment of latent TB is what is required to end the TB epidemic in the next 30 years. So the priorities for TB drug development are to have better treatments for drug-sensitive TB. Drug-sensitive TB is over 90% of the TB, as I showed you. and we need better treatments for that. We need much better treatment for multi-drug resistant TB because the existing treatments are terrible. We need to prevent drug-susceptible TB with shorter, safer regimens and prevent multi-drug resistant TB, which is a completely unmet need right now. So if you go back in history and look at clinical trials for TB, what were the endpoints that were looked for? Well, at the beginning, when antibiotics first became available, the major endpoint was survival. Did people live? And then clinical improvement. And then a lot of studies also looked at improvement of chest x-rays. So at the dawn of the antibiotic era, the British Medical Research Council performed certainly the most famous of the early randomized clinical trials. There's a debate about whether it was the first randomized trial, but certainly was the first prominent and important randomized clinical trial of the drug streptomycin, which had been discovered in New Jersey in the mid-1940s and developed into a drug. And this first randomized trial compared streptomycin treatment for TB to control, which was bed rest. Just lie in the hospital in bed, get fed, and don't do much. And what it showed after a year was that the streptomycin therapy reduced mortality by over 50%. Also had more patients who were clinically improved. And so this was evidence that streptomycin was effective treatment for tuberculosis and reminiscent in some ways of the early HIV therapy days of AZT. And like AZT for HIV, what they found was that there was a high prevalence of streptomycin resistance that evolved or was selected in patients treated with streptomycin. So the Medical Research Council went on to do a follow-on study where they gave streptomycin with paraamino-salicylic acid, or PAS, versus streptomycin alone, and they reported that that showed very similar clinical responses in the two groups, but they reduced the emergence of drug resistance from 70% with streptomycin alone to 9% with the combination therapy. So this was the first evidence for combination antimicrobial therapy to prevent the emergence of resistance. And this became a standard practice for all TB treatment thereafter. So that was in the 1940s, and through the 50s, a number of drugs were developed and studied in the 60s and 70s. And eventually we got from 1 to 2 years of treatment with streptomycin-based regimens down to a 6-month regimen of tuberculosis treatment. And how did we get there? The British Medical Research Council led a series of over 100 clinical trials of various combinations of drugs. And what they showed was that a 4-drug regimen given for 6 months was highly effective, led to very low relapse rates, and that treating longer than 6 months really didn't add value. So 6 months of treatment was established back in the 1980s. And these are the latest guidelines from the CDC, the American Thoracic Society, and the Infectious Disease Society, and they're all based on that same regimen from the 1980s. There have been some refinements, but basically we're using 1980s treatment for TB. Currently, there's been no change since then. But there's been a lot of work on improving treatment, and because mortality is no longer an important endpoint, because there's no need for anybody to die of tuberculosis, particularly in a clinical trial, we've had to evolve the outcomes that we look at. And so the outcomes we look for now are resolution of the illness, that people don't need to be treated, they're cured. And then an absence of microbiologic evidence of a recurrence, that there are no culturable tubercle bacilli following the end of treatment. So how do we get to new drugs for TB? You know, we have to do the preclinical discovery of compounds and all of the formulation, medicinal chemistry and toxicology work. But then step 3 is really key. We have to look at these agents in animal models that are predictive of outcomes in humans. And then we can do the early phase clinical trials looking at both pharmacology, and then phase 2 studies where we look at surrogate endpoints, things that we hope will predict longer term outcomes. So the two that have been, three rather, that have been used are all based on TB culture. So early bactericidal activity that measures the amount of killing of the TB bacillus that occurs during the first couple of weeks of treatment. We look at the 2-month culture conversion rate, and we look at the time to positive in culture. So many of you are now with COVID familiar with the cycle threshold. How many cycles of PCR does it take for the coronavirus to show up? Well, this is a similar type of measure. How long does it take in culture for the organism to replicate? And the more effective the treatment, the longer it takes to become positive. And then finally, it's not positive at all. When we have good signals in the early phase trials, then we can go to the large phase 3 clinical trials and look at clinical endpoints, such as cure and lack of infection. So we've been very fortunate here at Johns Hopkins to really have the world's leaders in developing new drugs for TB. Jacques Grosset is a professor emeritus. We got Jacques in 2001 when he was forced to retire in his native France because they said he was too old. But he came to Hopkins and for the next 17 years did some of the most magnificent work of his entire and long, illustrious career. Jacques had TB as a medical student in France and spent the rest of his career getting his revenge against the bug. And Eric Nürnberger, a professor of medicine here who has who trained under Jacques and has taken on the mantle of the leading experimental biologist for TB therapy, and their mouse model, the BALB/c mouse. Now, what Grosset had long ago showed was that this mouse model of TB recapitulated what we saw in human treatment, so that it cured mice with the 2-drug INH and streptomycin regimen in 18 months, and that's what was used clinically. INH and rifampin could cure this mouse TB in 9 months, and the addition of pyrazinamide led to curing patients, or mice rather, in 6 months, just as it does in humans. So this is a very good predictive model. So some of the first work that the group did was looking at new drugs that might be useful in TB, and the fluoroquinolones have good activity against TB, moxifloxacin in particular. And this is from the mouse, and it just measures the amount of TB in the lungs of mice over time, showing that adding moxifloxacin to the treatment accelerates the cure of TB in the mouse. Now, how do you take that into clinical trials? Well, I mentioned those many studies of the British Medical Research Council and Denny Mitchison, who led a number of those studies, published this letter to the editor in 1993 that suggested, and I apologize you can't see it, but I thought it was nice to show the original publication, that the culture conversion rate at 2 months was a very good indicator of the likelihood of a long-term cure after 6 months of therapy. And so we selected this 2-month culture conversion as our benchmark, as a surrogate marker for what would predict the longer-term outcomes for evaluating these drugs. And so we launched a number of trials of fluoroquinolones, of moxifloxacin given for 8 weeks. The first one was actually done by our team in Brazil, and the other next 2 were done by the TB Trials Consortium that Susan Dorman and I led this one, and Bill Berman from Denver and I led this one. And then Denny Mitchison, who was very active still, led a study in South Africa. And they all showed that moxifloxacin had increased killing power when given during the first 2 months of therapy, and speeded the time to culture conversion. not all of them showed a difference at the 8-week mark, which was our surrogate endpoint, but they all showed that there was faster killing when moxifloxacin was added. And so this led a number of groups to undertake phase 3 large clinical trials to shorten TB therapy by adding moxifloxacin or another fluoroquinolone, gadifloxacin, and all of these trials failed. None of them showed that the moxifloxacin containing regimens could shorten TB therapy from 6 months to 4 months. So here's one of them, the REMOX study, and this had 3 arms. 2 of the arms got moxifloxacin, and this shows that the moxifloxacin did speed the time to culture conversion significantly, although not dramatically, but it did not lead to a stable cure better after 4 months of treatment. These are the 2 moxifloxacin arms, and they were inferior to the control 6-month regimen. So that, and this is the OFLOTUB study, which used gadifloxacin, showing that the 2-month culture conversion actually wasn't better with gadifloxacin, and the overall favorable outcome was not non-inferior. They had a margin of 6% for non-inferiority, and I'll come back to that in a bit. So this led to a lot of recrimination and attacks against the mouse. Did the mouse mislead us? Were these, was this model wrong? So Eric Nürnberger and I and one of his postdocs went back and examined the mouse data very carefully, and this is the target. So the red is the control regimen, the standard regimen, and the blue is when moxifloxacin is added in place of isoniazid. And this is the goal to shorten treatment by 1 month, and the green line is the goal to shorten treatment by 2 months. And what happened in the mouse model is it was right in between. So the mouse model suggested that treatment could be shortened by 1 month with moxifloxacin, but not by 2 months. And so it became important to look more quantitatively at the animal model and not just say, oh, it's better, so therefore we can shorten treatment by a couple of months. Well, next there was a lot of focus on the drug rifapentine. Rifapentine is essentially rifampin with a slight chemical modification, but that modification makes it much more potent. It's 4 times as potent as rifampin, the cornerstone of TB therapy, and it has a much longer half-life. And so there were a number of studies looking at rifapentine, and this one led by Susan Dorman showed that increasing doses of rifapentine compared to rifampin led to much higher culture conversion at 2 months. And even though we know that the 2-month culture conversion is an imperfect surrogate endpoint, it certainly suggests that this rifapentine containing regimen should be very potent and perhaps able to shorten treatment. And then back to the mouse, when you combine rifapentine and moxifloxacin, you get much, this is looking at weeks, not months, much faster sterilization and cure of mice. So again, suggesting this would be a regimen that might be effective. So that led to this large 2,500 person clinical trial, the TB Trials Consortium Study 31, AIDS Clinical Trials Group 5349, that has just completed, where there's a control regimen, the standard 6-month regimen, and then there are 2 4-month regimens, one where high-dose rifapentine is given instead of rifampin, and one where both rifapentine and moxifloxacin are given for 4 months. Now, this study was completed, as I said. It's going to be presented in 3 weeks, 3 weeks from today at the International Union Against Tuberculosis Conference. So I can't give you any of the results, but I encourage you to stand by and watch with interest when these results come out. So to get to this study took 20 years. The first moxifloxacin studies were begun in 2001, and it's a long, long road. And the phase 2 trials take a good deal of time, and the phase 3 trials are very long and tedious and expensive. And so there's a lot of interest in improving the process. How can we get to these phase 3 trials faster? And this is some work that Patrick Phillips, a statistician now at UCSF, formerly at the Medical Research Council, has developed. And he's proposed the phase 2C study. So here's the classic phase 2B study for TB. You have a control regimen, and then you have 2 experimental regimens, or maybe just 1 experimental regimen, and you give the treatment for 2 months to look at that surrogate marker, and then you just put people on standard therapy and follow them for 6 months, but you're really only focusing on 2 months. And what Patrick has proposed is in phase 2C, give the treatment, the experimental treatment, the way you want it to be used in the phase 3 clinical trial. So here's the control regimen that goes out to 6 months, and then you follow them up for relapse. And here are the 2 experimental regimens, and you give them for 4 months or 3 months or 5 months or whatever it is you want to give. You treat them the way you want to in phase 3 and then follow them up. And when you do that, you'll have some outcomes. For the primary endpoint is the 2-month conversion, and you have very good power to detect differences in that. But it's only an intermediate endpoint, so it's a surrogate marker that's an imperfect surrogate. The secondary endpoint is the clinical endpoint of relapse, and it's an underpowered study, but it gives you a patient-relevant outcome. And what Patrick has shown through some simulations from those phase 3 trials that I mentioned is that, in fact, you have very good power to reject a regimen that really has a poor chance of succeeding in phase 3. And so he's proposed this as a much more efficient way to get to phase 3. Now, another approach that's been suggested is to do more individualized treatment, precision medicine, and that is to treat patients who are easy to treat with shorter regimens. And so it turns out that it's been done. John Johnson from Case Western did an early study. And he took patients who he said were easy to cure. That is, they didn't have cavities in their lungs, and their cultures converted at 2 months. And they got differentiated from the hard to cure patients. And the easy to cure patients, he randomized to get either a 4-month treatment, standard treatment, or 6 months, and it didn't work. And so that idea seemed doomed. But Rada Savic and her group, she is also at UCSF, did a much more intensive look at these easy versus hard to treat patients and found a number of predictors. So if you just look at the time of TB diagnosis, if you're a male, your likelihood of treatment failure is significantly greater. The higher your bacillary load, the more bacteria you have in the lungs and in the sputum, the higher the likelihood of treatment failure. And if you're HIV positive, you're harder to cure. And as you get older, you're harder to cure. You can also add some early on treatment outcomes to this and look at culture conversion, particularly the 2-month culture conversion. It turns out also BMI is a useful predictor. So you can put all these together and come up with tailored regimens for high-risk groups. And this is the SPECTRA trial that the TB Trials Consortium is currently developing, which is a stratified patient treatment trial using these baseline characteristics and some of the early treatment characteristics to distinguish those who might be easier to treat and can be treated with shorter regimens. Another approach that's being used by some groups is the MAMS trial design, the multi-stage, multi-arm, where you have a number of novel treatments that you're studying. You have a control group. And over a period of several years, you do a series of interim analyses and you reject the regimens that aren't performing well and aren't predicted to succeed in a phase 3 trial. And this was also something that Patrick Phillips was very involved in developing. I'm going to talk a little bit about multi-drug resistant TB. Globally, the outcomes of treatment for multi-drug resistant TB are just terrible. They're really awful. With long toxic regimens given for 1.5 to 2 years, only about half of the patients are actually successfully treated and cured. So this is an area of high need. A group from an NGO in Bangladesh developed a shorter regimen that used 7 drugs, existing drugs for TB, called it the Bangladesh regimen, and showed in observational studies that this regimen was as good as the longer regimen and obviously less toxic and cumbersome. So a randomized trial of this 9-month regimen was undertaken compared to a 20-month standard World Health Organization recommended regimen. And it was a non-inferiority study using a 10% margin for non-inferiority, and it used moxifloxacin. The Bangladesh study used gadifloxacin. And what it showed was that treatment success in both treatment groups was essentially the same, and the adjusted difference met the non-inferiority margin of 10%, and so this established non-inferiority. Now, when the results were first presented, in fact, because they hadn't finished all of the follow-up, but they presented it at a scientific meeting, they hadn't met non-inferiority. That upper margin of the difference was 10.1%. And then when they finished follow-up, it dropped to 9.5%. So that was a lot of controversy about whether they should have presented their results. And Patrick Phillips did this nice simulation of the STREAM study to question non-inferiority. As he said to me the other day, I'm a statistician who specializes in non-inferiority studies, which I don't believe in because it's so arbitrary. So here is one of the simulations he did. He said, what if the 9-month regimen was actually 3% better in this study? Would it meet non-inferiority? And the answer is, if you use a 6% margin, which is recommended by the FDA, no, it wouldn't. But if you had a higher margin, it would. What if it was 1% better, but with higher cure rates? Then it would meet all of the non-inferiority margins. What if it was 1% worse with lower effectiveness? Then it would only meet a large 12% margin. And then what if it's 3% worse? Well, depends. If you have a high cure rate, it meets a 10 and 12% margin. What if it's 3% worse, but a lower cure rate and doesn't meet any of them? And so what he asked is, given that 9 months is so much better in terms of patient commitment to the treatment, the cost of providing the treatment, and the toxicity of the treatment, would anybody not want to use a regimen that was within a 3% range of success? And it's likely that people would accept this. So choosing a non-inferiority margin is one of the big challenges. Fortunately, there are new drugs in the pipeline and actually in the field. There are 3 drugs now that have been approved for the treatment of MDR-TB, bedaquiline, nilamidine, and protamidine. These drugs were used in one of the most creative trials for treating highly drug-resistant TB, that is extensively drug-resistant TB, or TB that had failed, MDR-TB that had failed initial treatment. And this study was undertaken by Francesca Conradi from South Africa, and the TB Alliance based in New York. And they reasoned that because extensively drug-resistant TB, that's essentially resistant to everything, has no standard of care, that they could do a study with without a control group. And historical observational data showed that the mortality in XDR-TB was 80% at 5 years, and that basically it was impossible to cure very many patients. So they said, let's do a study of these novel new drugs, and let's not have a control group and just see how they do. And you'll notice it was published in the New England Journal of Medicine. So they treated patients for 6 months with these new drugs, the pretomanid and bedaquiline, and then they added linezolid, which is an existing drug, but novel for tuberculosis. They treated for 6 months. And what they've achieved was a 90% cure rate for MDR and XDR-TB. And there were some failures and there were some deaths. Most of the deaths occurred very early in the study. And so this was really a very impressive outcome for a novel regimen for what had been essentially untreatable TB. And on the basis of this study, pretomanid, which is one of the drugs in the regimen, was approved by the FDA. So a novel approval strategy, a novel regulatory strategy as well. Other approaches that the TB Alliance is pursuing is looking at a shortened regimen with novel drugs. This is bedaquiline, pretomanid, and moxifloxacin, and pyrazinamide for both drug-sensitive and drug-resistant patients. And the way they designed this trial is that this is randomized, and then the drug-resistant patients are simply recruited and treated with the regimen because we know that they require longer treatment and they don't want to power it for effectiveness. They simply want to be able to report good outcomes, assuming that the regimen is successful. And finally, another novel approach is the NTB clinical trial, which is led by Partners in Health and Medicine Sans Frontières. This is a study that's looking at 5 novel regimens based on the mouse work of Eric Nornberger. They're using Bayesian MAMS approach, where they're doing interim analyses and resetting their probabilities and dropping regimens along the way that don't appear headed towards success. So there's a lot of innovation going on with trials for treating active TB. So I come back to this slide just to emphasize that latent TB is also important. So I'm going to just quickly go over some stories of latent TB treatment. So George Comstock certainly was one of the most profoundly influential people in TB prevention of the last century. And George did a landmark study in Bethel, Alaska, in Alaskan natives in the late 50s and 60s to prevent TB with isoniazid. At the time, the prevalence of TB in this population was 2,000 per 100,000, so 2% of the population. The highest prevalence of TB in the world today is in Lesotho, and it's 900 per 100,000. So this was a terrible epidemic. 70% of children were infected with TB by the time they were 8. It was a very impoverished population. They had crowded living conditions, living in households that averaged 6 people with 1 room. And as Dr. Comstock wrote in his paper, it was a climate that discourages ventilation. So a very high-risk population. And he randomized using cluster randomization, and this was the first cluster randomized trial ever performed. He randomized the households for everyone in the household to get INH for a year or everyone to get placebo. And what he showed is that after a year, the TB incidence had fallen by 70%. And over the next 5 years, the overall reduction in TB incidence was 60%. But what you'll note, and what's important about this cluster randomized trial, is that there were also dramatic reductions in the placebo arm over this time period because they had the secondary benefit of not having TB transmitted by these people who got the INH. So this showed both the community level impact of INH preventive therapy, but clearly demonstrated that a year of INH preventive therapy was effective. Now, Dr. Comstock, and this was before the field of bioethics existed. It was before the Belmont report. There was no such thing as an IRB. But Dr. Comstock said, these people, sacrificed to be in this study. They donated their time, their effort, and their lives to finding this answer. They all deserve treatment. But the Public Health Service wouldn't fund him to continue on the treatment. So he just went back and he gave everybody INH for a year, reasoning that, well, maybe another year will be better than a single year. And that allowed him to do a retrospective observational analysis, say how much INH is needed. And what he showed is the longer duration of INH, the better until you got to 9 months. And then after 9 months, there really wasn't much improvement. And so 9 months was the optimal duration of INH preventive therapy. And that became the US standard. But the problem with INH preventive therapy is it can cause liver damage, it can cause other toxicities, and the adherence is poor. In the US and Canada, it's estimated that only about half of people who get put on INH preventive therapy actually complete it, and there's been low uptake around the world. So we need better, shorter regimens to improve use of preventive therapy. Now, if you go back to this pathway to new regimens, what you see is that there's no surrogate marker for latent TB. So what we end up having to do is go from mouse models to phase 3 trials. So that's a big leap of faith. But that's what we have to do. Now, fortunately, Dr. Grosset and Nurnberger developed a nice latent TB model in the mice. They first immunize the mice with BCG and then infect them. And what happens is with the immunization, the mice have a low-grade TB infection that is stable, it's not lethal. If they don't get the immunization, the infection takes off and is lethal. But this then allows them to study experimental regimens to kill these low-level replicating bugs. So it's a model for humans that has proved to be highly predictive, highly effective, even though it's not pathologically the same disease state. So some early work that Dr. Grosset did, looked at the drug rifapentine with or without INH in this model, given for varying durations. But with the rifapentine given for 13 weeks, 3 months, was highly effective. And so that led to 3 clinical trials of this rifapentine INH given once a week to people with latent TB. And this regimen is abbreviated 3HP, 3 months of H for INH and P for rifapentine. The first one we did in Brazil in household contacts of TB patients, the second one in HIV-infected people in South Africa, and the third one a large trial in the US, Canada, Brazil, and Spain in high-risk patients. And what all 3 of these studies showed was that the 3HP was non-inferior to isoniazid, and it was safer. It had less hepatotoxicity, better treatment adherence, and treatment completion. And this just shows the largest study, the TB Trials Consortium Study 26, where the 3HP regimen was within a whisker of being significantly better than the 9 months of isoniazid. And then when in a sub-study of people with HIV, again, the 3HP was better, not significantly better. It was clearly non-inferior, and so this has become a standard of care regimen. And one of the reasons that it's better is that people finish it. The shorter the period of treatment, the less opportunity there is to give up. And what you can see in study 26 is a lot more people gave up on INH because they had more time to get tired of it. Not sick from it, just tired of it. And that's the main reason that people stop, is they're tired of it. Well, at the time we were finishing up these trials, Eric Nurnberger said, can we do better? What if we give the rifapentine and INH daily for a month instead of weekly for 3 months? And in the mouse, that looked very good. So that led to this large phase 3 clinical trial of 1 month of rifapentine and INH, or 1-HP, in people with HIV that we undertook with the AIDS Clinical Trials Group. So a 3,000 person study, all with HIV, adolescents and adults who had no active TB, and 97% of them came from high burden countries. We did tuberculin testing at baseline to see if they had documented latent TB, and only a quarter of them did. However, we know from many other studies that people with HIV who have a negative tuberculin test still have a very high risk of TB compared to HIV negative people. So the WHO recommends that all HIV people, positive people in high burden countries, receive preventive therapy. And so this is the treatment, the daily 1-HP versus the control of 9 months of isoniazid. And this is the outcome. Clearly effective and clearly non-inferior. And when we look at that subset of people who were positive for latent TB, they had a positive tuberculin skin test, PPD, or an interferon gamma release assay, we see that the point estimate is a little better, but the non-inferiority margin is easily met. And this is now a new recommended regimen by the World Health Organization. So we've gone from 9 months with Dr. Comstock's work in Alaska, originally a year, but then 9 months, and then we got it down to 3 months, and now we have it down to 1 month. So are we done? Well, of course, Eric goes back to the lab and says, well, can we do better? So the first thing he found in the mouse model, that here's the 1-HP regimen in an experiment, and right next to it is bedaquiline, J standing for Johnson & Johnson, who make bedaquiline. These drug abbreviations are somewhat idiosyncratic. And 1 month of bedaquiline is just as good as the 1-HP regimen, but it would treat MDR TB. What happens if you add rifapentine to bedaquiline? It's even better. So maybe this is a 2-week regimen. What happens if you give 3 drugs, pyrazinamide? Well, then, this could be a 1 to 2 week regimen. So we're not there yet. We really might do better. And bedaquiline certainly is appealing because of its potential for multi-drug resistant TB. Well, there's been a lot of interest in long-acting injectable drug formulations for HIV recently, and some big successes. So Johnson & Johnson, working with Eric, have been looking at long-acting bedaquiline injections. And this is the PK of this in a mouse model. And a single injection of this long-acting formulation of bedaquiline in mice leads to very active levels of both the drug and its primary metabolite, which is somewhat active against TB, for 3 months. So a single injection provides coverage for 3 months. And Nicole Ammerman and Eric have done some studies in infected mice and given 1 shot, 2 shots, or 3 shots over a 2-month period. And what, I don't know if this is projecting well, but at 3 months, all 3 of them are very effective. Even a single injection is leading to great reductions in the bacillary load in the mouse. So very promising future for long-acting injectables. So I'm just going to wrap up here, leave time for some questions with a few concluding thoughts. Regimens to treat and prevent TB have come a long way since that first streptomycin trial, but we're not there yet. We need better regimens both for active TB and for latent TB if we want to control the TB pandemic globally. TB trials have been a source of innovation in clinical trials since the very first BMRC study, the first big randomized trial in history. And Dr. Comstock did the first cluster randomized trial in history. And you've seen a few ideas for new innovations in trial design with MAMS, a trial of XDR-TB with no control group that leads to an FDA approval in a New England Journal of Medicine paper. And lots of other innovations. So the study of TB has really helped spur some of the most important developments in clinical trials design of our lifetime. And future innovations are going to help achieve the NTB targets of the United Nations to eliminate TB in our lifetime. So I'll end and thank a number of colleagues who contributed to the work that I've showed you, particularly Eric Nurnberger and Jacques Grosset, Nicole Ammerman, other colleagues, Susan Dorman, who's no longer with us, who's at the University of South Carolina now, Sue Swindles from the ACTG, Tim Sterling, our former faculty member now at Vanderbilt, and a number of colleagues from the TB Trials Consortium, and Patrick Phillips, who shared some of his really exciting ideas that I showed you, and the funders that I've had for much of this work. And I will end there. I'm happy to take any questions.
So Dick, that was spectacular. I thought it was a terrific talk. I'll start with the first question. Recommendations for extrapulmonary TB include longer treatment durations. Are there data to support that, or is that just, we think it's harder so we treat longer and the treatment stuff is arbitrary?
Well, the treatment of extrapulmonary TB, generally the recommendations are to treat for the same duration, 6 months, with 2 exceptions: central nervous system disease and bone and joint TB. And the rationale there being that it's harder to get the drugs in there. So getting into the brain, getting into the spinal cord, maybe getting into the eye, the central nervous system part of the eye, and getting into bone. But for other forms of extrapulmonary TB, it appears that 6 months of treatment is very effective. And that's actually in the guidelines.
So, but the neuro TB and bone and joint TB, do we have good data, or are they really just arbitrary because we don't have enough data to really make evidence-based recommendations? I mean, I understand the rationale for them, but.
We have some data. There have been some controlled trials in some of the forms of extrapulmonary TB. For central nervous system TB, the controlled trials have largely been looking at the addition of steroids or other immunomodulatory therapy as a way to improve outcomes. And the rest of it is just based on clinical experience and expert opinion. Those are very hard studies to do. Kelly Dooley is currently leading an NIH-funded study for treating TB meningitis in children. And it's exceedingly difficult to recruit people for that study. They're just very hard studies to do because it's a fairly uncommon condition. Most of the recent studies of TB meningitis have been done in Vietnam by a group from Oxford working in Ho Chi Minh City, and they've been able to recruit patients, but it's very difficult to do, so hard to study.
Okay. We are open for other questions.
Hello, can I ask a question? Could you please comment on liver toxicity regarding the drug combination, and what are the treatment concerns for those susceptible patients, and like how would the trial results be applicable to them?
So liver toxicity is probably the major concern with TB treatment, and we know that a number of TB drugs have liver toxicity. The most famous is certainly isoniazid. It's very interesting that isoniazid, was developed in 1952. Dr. Comstock used it in Alaska. The U.S. Public Health Service did studies in over 100,000 people. And there was never a case of liver toxicity reported until the late 1960s. There was an outbreak of TB in the U.S. Senate, and a number of Senate staff were put on INH preventive therapy, and there were a couple of deaths from INH hepatotoxicity, which was a new phenomenon. People were surprised by it. But it clearly happens, and why it wasn't reported before is a bit of a mystery. But it's certainly the most famous hepatotoxin. Pyrazinamide is actually even more hepatotoxic. And then rifampin is somewhat hepatotoxic. So you've got 3 drugs that can damage the liver. And so it's a great concern. And so in clinical practice, as well as in clinical trials, people are monitored for liver toxicity, both clinically and with lab studies. Of the new drugs that have been studied, only one, pratominid, appears to have had any hepatotoxicity, but it's not clear that it's really the culprit. But in a study that we did with the TB Alliance, we had 3 deaths from hepatotoxicity in people receiving pratominid, but they were also receiving pyrazinamide. But we didn't have any deaths in the control group that was receiving pyrazinamide. So it's an early warning signal, but it hasn't been seen in other trials, fortunately. But it's a major concern, and so every single trial has liver monitoring as a central safety component.
Thank you.
Any other questions? So Dick, I think you've made it so clear nobody has any questions, which is always an amazing
I have
sight.
another question.
Okay, great.
So thank you. We noticed that a lot of those TB trials have been conducted in underdeveloped countries. What are your experience that you could share with us when conducting trials in underdeveloped countries, like in terms of logistics, like data integrity, practice, things like clinical practice, things like that?
Yeah, so I mean it's an important question. Shortly after I arrived here in Baltimore, I was asked to take over the Baltimore City TB clinic because they didn't have a director, and so I did, and brought a team from Hopkins in there for the first time, and we started doing clinical research. And there was some tension between the city employees and the Hopkins employees about what our goals were. So I had a retreat, and we all got together, and I asked everyone to tell me in 5 years, what headline do you want to see in the Baltimore Sun about TB in Baltimore? And one of the old time, workhorse city nurses, who was an academic skeptic, said, the headline I want to see is TB eliminated in Baltimore, Hopkins researchers forced to go overseas. And I said, I want to see that too. And we haven't eliminated TB in Baltimore, but we pretty much made it impossible to study. And it's pretty much impossible to study active TB in the United States because we have about 9,000 cases a year, but they're distributed all over. And so if you want to study, an important disease, you go to where the disease is. And, TB is a very important disease. It's just very common in high burden countries, which tend to be low or middle income. So that's the first point, that you go there because that's where the disease is. But it's important. I've always found it important to have local partners there, not, an international team coming in, doing a study and then coming back and, leaving wreckage in its wake. So I've always worked with local partners on the ground who are connected to the TB program and who, we develop capacity with so they can become TB researchers. One of the, in my slide here, Neil Martensen, for instance, is a South African physician who went from working for the local health department in Johannesburg to coming to Hopkins, getting an MPH, and now he's the executive director of the perinatal HIV research unit and an adjunct professor at Hopkins. And that's just an example. Marcus Conde is a professor at the Federal University of Rio de Janeiro, another colleague. So building capacity on the ground is essential, and doing the research in the context that's relevant. We don't want to go in and do studies that are completely irrelevant just because they're academically interesting to us. We want what we study to be relevant, and then deliver what we've discovered to the country and to the world. And so all of the studies that I've talked about, then go to the WHO for consideration of how will this be implemented in practice globally. So the 1HP study that I showed you, we published last year, and earlier this year, the WHO recommended that as a standard first-line regimen. The 3HP we published a few years ago, and the WHO has recommended that. So, and I'm currently working on a project funded by Unitaid to increase the uptake of TB preventive therapy with rifapentine in 12 low and middle income countries, because they recognize that these things just don't happen. A New England Journal paper doesn't mean that a country is going to start delivering a therapy. There has to be a process to implement, and so we're working on that as well.
Thank you very much.
You bet.
Can I ask another
Okay,
question?
well, Dick, thanks an awful lot. This is a spectacular talk. We appreciate it greatly. I think we'll wrap it up now, and I'm sure we'll ask you back in a couple of years and you'll tell us that you've achieved your goal of eliminating TB.
Well, we'll see, but thank you very much. Really appreciate your having me. So thanks.
Thank you.
All right.